Thank you very much, Jeremy, for the invitation and the introduction. And what I'd like to do today is walk you through some of the work that we started at Washington University and that we continue at Penn, designing cancer vaccines that are personalized, because we use passenger mutations that are present in the tumor to activate the immune response. So I'll talk to you a little bit about the vaccine, the design of the vaccine, what immunity the vaccine elicited, and then also about the characteristics of the T cells that come out of the vaccination. So I have worked in cancer immunology for over 15 years, and I think it has been in the last 5 to 7 years that really we have had important advances that have shown us that we can harness the immune response to treat cancer. And most of these evidence have come from two places. One is the development of checkpoint inhibitors. These are antibodies that target either PD-1 or CTLA-4 in the surface of the cells, and that by blocking the interaction of the receptor and the ligand, we can enhance immune responses to the tumor. Their FDA approval and their use in the treatment of melanoma, lung cancer, and colon cancer have shown then that we can activate the immune system towards cancer. And then the second evidence is more recently from adoptive Tcell therapies, where we can again use T cells to eliminate and reject the tumor. But although these are striking results and have really supported the study of the immune system in cancer, not everybody responds to these treatments. So this is a figure actually from Anthony Rivas, from an Anthony Rivas paper, where he looked at patients that had and had not responded to anti-PD-1 therapy. And what he saw was that in the patients where anti-PD-1 therapy worked, you had a presence of CD8 infiltrating the tumor. The tumor also expressed the PD-1, and the tumor and the T cells within the tumor expressed PD-L1. So it made perfect sense here that we could use an antibody to target this receptor-ligand interaction to activate these cells and get an immune response to the tumor. And he named these tumors hot tumors. On the other hand, you have patients that are like this, where there is very little infiltration of T cells and little expression of PD-1 or PD-L1. So we refer to these tumors as cold, and these patients usually progress. So of course then the idea is, can we take patients that present this kind of a scenario, and can we vaccinate it so that we can either initiate or reinvigorate a response against the tumor? So the question also always has been then in cancer vaccines, what do we target the immune response to? Until recently, most of the response have been to these self-antigens, to these share antigens, which tend to be proteins that are shared between patients. But we knew also that we could use passenger mutations from data that was generated in the 1990s, but it was difficult to know how we could, in a timely manner, identify those mutations and make a therapy. So fortunately, technology has advanced, and now we have next generation sequencing, which not only allow us to identify the mutations that are present in the tumor, but allows us to do this in a digital manner. So we not only can know which mutations are present, but also we can know at what frequency and how many of the tumor cells present a given mutation. And when you look at, this is a very famous figure that everybody presents, where we're showing is the mutational burden for the various tumors. What we see is that melanoma, it's the highest mutational burden among tumors. And the reason for that is because most of the mutations that we find in tumors in melanoma arise from UV radiation. So they tend to be passenger mutations, and they're usually C to T transitions, which is characteristic of UV radiation. So we decided to start a clinical trial on melanoma, designing a vaccine that would target those missense mutations. So we started this project in about 2013, 2012, 2013. The observation, as I said before, was one from Terry Boone's lab and the other one from David Beach's lab, where they had actually shown that missense mutations can elicit T-cell immunity in humans with cutaneous melanoma. And at the time when these studies were done, they were using cDNA cloning to identify the missense mutations, and it took them about a year from the time they started the project till the time the mutation was identified, and it was one mutation in each patient. So we hypothesized then that we could use single nucleotide variants that encode amino acid substitutions to generate patient-specific tumor neoantigens, and that then we could elicit responses to these neoantigens by immunization. So we used next-generation sequencing then to identify the mutations, and we generated a pipeline that took us from the genomic data to the postulate of the predicted neoantigens. And then we vaccinated patients and asked the question, does vaccination using missense mutations increase the breadth and the diversity of the anti-tumor response? And the experimental approach that we used was to use autologous dendritic cells as a vaccine. And the reason for that is because we had spent 10 years previously devising a protocol where we had shown that if we took autologous dendritic cells, we matured them with a combination of 4 activation markers, that's CD40 ligand, interferon gamma, TLR3, and TLR8 agonists. We could really induce these cells to make IL-12, and that the production of IL-12 was correlated with clinical outcome. So we took these autologous dendritic cells in, and we used them pulsed with peptides that had contained missense mutations, and then we went into the patients, vaccinated, and wanted them to characterize the responses that the vaccine elicited. So this is basically the scheme that we use to identify these missense mutations that then lead us to make amino acid substituted peptides that will incorporate the vaccine. So we basically take DNA and RNA from the tumor and DNA from PBMCs, and we do sequencing. Then the exome sequencing data that comes from the normal DNA and the tumor DNA is used to identify variants, so to identify the missense mutations. Then these missense mutations then are translated into amino acid substituted peptides that then are run through a series of algorithms that make predictions of which ones will bind to the patient's HLA haplotype. And then we integrate that information with the RNA so that we know that these mutations are coming from messenger RNAs that are expressed. We take the allelic frequency, the expression, and the immunological information, and then we pick candidates. And then from those candidates, we synthesize somewhere between 7 to 12 peptides that then are confirmed to bind to the HLA of the patient by in vitro binding or by HLA refolding. And then 10 to 12 of those patients then are formulated into a vaccine. So we have a pipeline that is called PVAC-seq that has been published to carry all this process. And then what we do is then we incorporate it in a vaccine. The vaccine consists of 3 doses of mature dendritic cells that are separated by 6 weeks between them. We leukophorese the patient before the vaccination, so these leukophoresis acts as a source of dendritic cells for the vaccine. The patients get Cytoxan to modulate the Tregs that are present in the patient. Then fresh dendritic cells are made, and 150 million dendritic cells are given in dose 1, approximately 15 million per peptide. And then 6 weeks later, they get a second dose of about 60 million, 5 million per peptide, and a third dose. And then the patient is leukapheresis again, and this is a research leukapheresis so that we can understand what kind of immunity the vaccine elicits. And during all these process of vaccination, we collect blood weekly, and that blood then is used to do immune monitoring. And the immune monitoring that we do is usually take phycohypec isolated PBMCs that are cultured in vitro in the presence of the different peptides and IL-2, and then we do a multimer assay to see how these respond, if and how this response is developing. And most of these has, for 3 patients, have been published. So I just show you our immune monitoring, which requires the cells to be expanded ex vivo. So I always get asked, well, that's expansion in vivo. Can you detect the neoantigen T cells directly from the blood? So I'm just showing you here one patient. This patient actually was vaccinated with 7 mutation-containing peptides and responded to 3. And what I'm showing you here is before the vaccine and after the vaccine. These are phycohypec PBMCs that are then being stained with a multimer. And what you see is that for both the neoantigen, there was very incipient, if any, response prior to vaccine, but then after the vaccine, about 1% of the cells actually are directed towards the neoantigen. This patient also got 2 shared neoantigens, GP100 peptides, again, very little an enhancement of the response after the vaccine. And then I'm just showing you here a flu response that did not change, which is what you would expect. Are these responses long-lasting, or are they short memory cells that are being elicited by the vaccine? So here is data again from another patient. This patient also had 3 neoantigen responses that were elicited by the vaccine. And you can see that after the 3rd dose, you can detect these cells after in vitro culture very clearly. You can come back 6 months later, and now this patient has been disease-free for over 3 years, and you can still expand these neoantigen-specific cells from the blood. So it appears that the vaccine elicits a long-lasting immune response that leads to memory. When we look at affinity, whether the affinity is predicted, and I'm just showing you one of the algorithms here that we use to predict affinity, it does appear that the ones that are immunogenic have higher predicted affinity than the non-immunogenic. However, there is a large group of non-immunogenic here that have affinities below 50 nanomolar. When you do experimental binding, which we always do for the peptides before we include them in the vaccine, because we want to make sure that at least we're targeting something that can be presented by the patient's HLA. Again, you see that the experimental affinity for the immunogenics is higher than the non-immunogenic. But still there is a small group here that has high affinity and are not immunogenic. So, and then when we look at stability, again, this is another predicted stability based on Sorenbos model. We see that what is clear is that the more stable the complex is formed between the peptide and the MHC, the more immunogenic you're likely to be. This is still a good stability, and these peptides are non-immunogenic. So we still have a lot to learn as to how we can go about picking what is immunogenic and non-immunogenic. We also have a lot to learn about how we can not use cryptic peptides in our assays. And for this, we're going to start incorporating proteomics as part of the selection mechanism. So what have we learned then from these first 5 patients that have been treated with the neoantigen vaccine? We are starting to learn about the immunogenicity. What we see is that in each of these patients, there is always some kind of pre-existing immunity. The patients then, at least for us, the ones that we have studied, there is always 1 or 2 pre-existing responses to mutated peptides. Then the vaccine elicits responses to additional neoantigens, but that there is about 50% of the peptides that we use that are non-immunogenic. What is characteristic of the immunogenic peptides is that these tend to be high affinity, both experimentally and predicted. They tend to be less than 50 nanomolar. So now, if it's possible, we only pick things that are 50 nanomolar or less, which you cannot always do, but that's the goal. And then when we look at these peptides and we ask ourselves, what are there any positional bias? Are the mutations more likely to be in the anchor residues, or are they in the residues that point up towards the T-cell receptor? There is really no positional bias. We have gotten immunogenic peptides that the substitution has been in an anchor residue, and we have got immunogenic peptides that are in the T-cell receptor contact residue. So that doesn't, so far, there appears that either one can elicit responses. One of the nice things about the vaccine is that it increases the antigenic breadth. We have 1 or 2 pre-existing, but then we immunize and we see more reveal. We think this is important because by increasing the antigenic diversity, you might address the tumor heterogeneity. The more antigens you target, the less likely it is that there will be tumor escape. And then in terms of the clinical response, of the 5 patients, 3 patients are alive and are in remission. I told you 2 of them just got the vaccine. Now this is 5 years, this is 3 years. And then one of the patients went into PD-1 therapy, and now it will be coming up to 3 years since it has been free of disease. So let me now change a little bit and talk about the neoantigen-specific cells themselves. So we're very interested in understanding the clonal diversity of the T cells that are elicited by the vaccine. And this is based on the fact that we know that in viral diseases, having a clonally diverse, so having many T cells that have different TCRs directed at a given antigen, it's supposed to provide a certain degree of fitness. So we wanted to see if these neoantigen-specific cells were diverse or not. So the way we went about doing this was by using next-generation sequencing, targeting the T-cell receptor, the TCR beta portion of the T cell, and this was done using the adoptive biotechnology. So basically, you use a set of primers that target the TCR beta. And what you see, and we then used a scheme where we sorted the cells, and then we did next generation sequence. We made a library and went back into the pre-vaccine, post-vaccine, and the tumors, and we look at the T-cell repertoire. Interestingly, if you go and look then in the blood, what you see is that in the blood, the diversity is higher, and that then if you vaccinate, that diversity becomes even higher. So you go from 1 to 9 clonotypes before the vaccine, and then that goes to 20 to 5 clonotypes after the vaccine. So the vaccine really enhances the diversity. So we think that the endogenous pre-existing immunity is characterized by very little diversity. You find 1 to a few, 1 in the tumor, a few in the blood. The oligoclonality is a feature of the tumor infiltrating cells, and then when you vaccinate, you recruit new clonotypes, and then we're interested in studying whether that diversity is a measurement of fitness. And then we also want to see when then the patient goes into anti-PD-1 therapy, what happens both to the antigenic breadth and the TCR clonotype diversity. Then I'm just going to acknowledge those that did the work, our group at Penn, the research group, and also the manufacturing group for the vaccine, and our collaborators for the T-cell receptor portion. Our previous lab manager at Washington University, our molecular genomic group, Elaine Martis. We started this collaboration with Elaine when we were both at WashU. Now she has moved to Children's. David Spencer, with whom we continue the collaboration, the genomic portion, and our funding agencies. Thank you very much.